Hello everyone, this is LATHA AKULA. In this video we are going to study about another topic in biomolecules, that is lipids. Here these lipids are made up of carbon, hydrogen and a little amount of oxygen. And here these lipids are not soluble in water. We know that if we add any oil or any lipid in water, that will not soluble, hence they are water insoluble. Of course they are water insoluble, but they are dissolved. If they were dissolved in inorganic solvents, they can be easily dissolved. Examples for those inorganic solvents where the lipids can be dissolved is ether, benzene and acetone. And next, here these lipids are not polymers and these are not strictly biomacromolecules. Lipids are not strictly macromolecules. Their molecular weight does not exceed 800 daltons, but they come under acid insoluble fraction because when we grind any tissue or cell membrane and any other membrane, that will be broken into pieces and form vesicles, right, which are not water soluble. So here therefore these membrane fragments in the form of vesicles, they get separated along with the acid insoluble pool, hence they were found in the retinate and included in the biomacromolecules. These are not strictly biomacromolecules like the other biomacromolecules. And here these lipids are esters of fatty acids and alcohol. What they contain? They consist of esters of fatty acid and alcohol. And here the formation of lipid is called as a lipogenesis. Here what is the meaning of genesis? Genesis means synthesis and production. Lipogenesis means the formation of lipid, and whereas the breakdown of lipid is called as lipolysis. Lysis means breakdown, right? And if we take 1 gram of the lipid, we'll get 9 kilocalories of energy. How much energy we'll get from the lipid? So we'll get nearly 9 kilocalories of energy. And for example, if we have taken 30 grams of the lipid, how much energy we'll get? That is 30 grams is multiplied with the 7 and we'll get 270 kilocalories of energy. And this is about the introduction part of the lipids. If we have taken the classification part, the lipids can be categorized into 3 groups. How many groups are found? These lipids are categorized into 3 groups. They were simple lipids, compound or conjugated lipids and derived lipids. So how many types are there? There are 3 types of lipids were found. They were simple, compound and derived lipids. And again this simple compound lipids have been again divided into the simple lipids. They have 2 subgroups. They were neutral or true fats and second one is the waxes, whereas the compound or conjugated lipids again subdivided into phospholipids, glycolipids, lipoproteins and chromolipids. What are they? Phospholipids, glycolipids, lipoproteins and chromolipids. And whereas in derived lipids, examples for those derived lipids are sterols, terpenes, prostaglandins, all will come under the derived lipids. So in that the first one, what is the first one? Simple lipids. What is the first one? Simple lipids. So in that simple lipids, if we take what are the simple lipids exactly, we need to know. These are nothing but the esters of the fatty acid with various alcohols. So what are the simple lipids means? These are the esters of fatty acids with various alcohols. So again simple lipids have been subdivided into neutral fats, neutral or true fats and waxes, right? In that the first one is neutral or true fats. So what are the neutral or true fats? These neutral or true fats are nothing but esters of fatty acid with glycerol. Fatty acid with what type of alcohol it is? It is glycerol. And another name of these neutral or true fats is they are also called as glycerides. What are glycerides? Glycerides are nothing but neutral or true fats. And here if we take a fat molecule, what it contains? If we take a fat molecules, it contains 1 molecule of glycerol and 1 to 3 molecules how many? It consists of 1, it may be 2, or it may be 3. So 1, 2, 3 molecules of same type, or it may be different type of fatty acid. What the fat molecule consists of? One molecule of glycerol is present. It may contain either one molecule of fat, or 2 molecule, or 3 molecules of same type of fatty acid, or a different kind of fatty acids will be present in a fat molecule. So we have studied that the fat consists of glycerol and 1 to 3 molecules of fatty acids. So now we'll see glycerol. What is the glycerol? It has 3 carbons are present. So what the glycerol consists of? It has 3 carbons. See here, this is the glycerol structure. How many carbons are present? 1, 2, 3 carbons are present, and attached to this hydroxyl groups. So 3 hydroxyl groups are present. This is the structure of a glycerol. Now we'll see the fatty acid. So what is a fatty acid? It is an unbranched chain of carbon atoms. They have carboxyl groups. What they consist of? Carboxyl group is present. Attached to this, there is an R group. A functional group, R group or functional group may be present. So this R group may consist of either a methyl group, ethyl group, or they may consist of CH2 groups are also present. Here the number of carbons may ranges from 1 carbon to 19 carbons may be present. If we take some examples for the fatty acids, we will see first example is the palmitic acid. Here this palmitic acid, it consists of 16 carbons including carboxyl group. Including carboxyl group, how many carbon atoms were present? 16 carbons. If we have excluded carboxyl group, carboxyl carbon, how many carbon atoms will be present in palmitic acid? 15. And the second example is arachidonic acid. Arachidonic acid, including carboxyl group, it consists of 20 carbon atoms. If we have excluded carboxyl group, it consists of only 19 carbon atoms. And here these fatty acids are of 2 types. How many types of fatty acids were present? 2 types of fatty acids were present. The first one is saturated fatty acids. Here what is meant by saturated fatty acids? They consist of a single bond. The saturated fatty acids, they consist of a single bond. Examples for these saturated fatty acids were palmitic acid and stearic acid. So this is the palmitic acid. We need to remember the examples of the saturated and unsaturated. Not only that, we need to remember the how many carbon atoms present. So how many carbon atoms were present in the palmitic acid? 16. See here there are 14 and 15 carbon, and including carboxyl group, 16 carbon atoms were present in the palmitic acid. And if we see stearic acid consists of 18 carbons. See here, how many carbons are present? 16, right? And 17. This is 17, and including carboxyl group, it has 18 carbon atoms in stearic acid. And another example for the saturated fatty acid is arachidic acid. We need to remember what is this? This is arachidic acid. It consists of 20 carbon atoms. How many? 20 carbon atoms. So what are unsaturated fatty acids? They consist of 1 or more double bonds. How many? It may consist of 1 or 2 or 3, anything. So 1 or more double bonds are present. They are called as unsaturated fatty acids. Again, these unsaturated fatty acids are 2 types. They were monounsaturated fatty acids. In short, they can be called as MUFA. And polyunsaturated fatty acids, in shortcut we can call it as a PUFA. MUFA and PUFA. So monounsaturated fatty acids, they contain a single bond. How many? Single double bond is present. Only one double bond is present. They will come under monounsaturated fatty acids. Examples for these monounsaturated fatty acids were oleic acid. What is this? Oleic acid. See here, this is the formula of oleic acid. How many double bonds were present? Only one double bonds were present. And next one is polyunsaturated fatty acids. They consist of more than one double bonds. Here contains more than one. We have studied saturated fatty acids, they consist of single bond, whereas unsaturated fatty acids, they consist of double bonds. And again, depending on the number of double bonds, they were classified into two types. If there is only one double bond, they will come under MUFA. If there is more than one double bond, they will come under the PUFA. And here, examples for these polyunsaturated fatty acids were linoleic acid. How many double bonds were present? 2. Linolenic acid, it consists of 3 double bonds. And arachidonic acid, we need to remember this is arachidonic. Already in the saturated fatty acids, we have studied about arachidic acid, right? Arachidic acid, that will come under saturated fatty acids. We need not to confuse. Arachidic acid is a saturated fatty acid, and arachidonic acid will come under the unsaturated fatty acid. It consists of 4 double bonds. So here, this is a linoleic acid formula. How many double bonds were present here? How many we have written? 2. See here, this is 1 double bond, this is second double bond. 2 double bonds were present in the linoleic acid. Linolenic acid consists of 3, and arachidonic acid, it consists of 4 double bonds. And these polyunsaturated fatty acids, when compared to the monounsaturated fatty acids, they were easily digestible. As where they were present? They were present in the oils of sunflower and safflower. And these polyunsaturated fatty acids, they were good for the heart patients. In the neutral fats are true fats. They may be monoglycerides. That means if they consist of only one molecule of fatty acids. So that means glycerol with only one fatty acids. We have already studied about the fat molecule, they consist of one glycerol, and it may consist of 1 to 3 fatty acids, right? If depending upon the number of fatty acids attached, these neutral fats were classified into 3 types. There were monoglycerides, diglycerides, and triglycerides. Monoglycerides means if they consist of only one molecule of fatty acid attached to the one molecule of glycerol, that those are called as monoglycerides. If 2 molecules of fatty acids attached to the one molecule of glycerol, here we need to remember the glycerol is only 1. The number of fatty acids were changing. 2 fatty acids here, 3 fatty acids were attached. It is a triglyceride. Here the glycerol is 1 only. And if you see structure of this is the glycerol structure, right? It consists of 3 carbons and 3 hydroxyl groups were present. These are the fatty acids. This is the first fatty acid, second, third. For example, if the glycerol is attached to the one fatty acid, here what happens? This is the R group, this is carboxyl group, right? Here CH2OH. Here this hydroxyl group in fatty acid and hydrogen in the glycerol, they will be removed in the form of a water molecule, dehydrated. And what will happen here? See, there is an ester linkage is created between the glycerol and fatty acid. What is this linkage? Is called ester linkage. If it is 1, and if a second fatty acid is also attached, it is a diglyceride. If a third fatty acid is also attached, then it will form the triglyceride. See, this is the triglyceride. And from the triglycerides, how many water molecules were removed? 3 water molecules were removed. See here the number of glycerols, only 1 molecule of glycerol only, but here the number of fatty acid changes. It may be 1, it may be a 2, it may be a 3. If 1 fatty acid is attached, it is a monoglyceride. 2 fatty acids were attached, it is a diglyceride. And 3 fatty acids were attached, it is the triglyceride. And what is the linkage between the glycerol and fatty acid? That is ester linkage. What is this? That is ester linkage. Here, these waxes are waterproof in nature. That means allow the water to enter in. Here, these waxes are the esters of fatty acids with alcohol. Instead of glycerol, what is present in this? alcohol is present, that too high molecular weight alcohol is present in the waxes. Examples for these waxes were, example, beeswax. This beeswax is called hexacosyl palmitate. Chemically it is called hexacosyl palmitate. And what is this beeswax? We know honeybees, right? That produce honey. And there are different types of honeybees in that. Some are queen bees, some are drones are present, some worker bees are present, right? In that, the what is the house of the honeybees is called beehive. How that is produced? By means of this beeswax only they build the beehives. And this beeswax is produced from the abdominal glands of the worker bees. The abdominal glands of the worker bees, they secrete this beeswax, and these worker bees, they build the beehive. And second example is the cerumen, which is called as earwax. In our ear also there is a wax is present, right, which is called cerumen. And what is its function? It prevents the entry of dust particles to enter inside. That is, it traps the dust particles in the ears. And third example is wool fat. Here, wool, we know that sheep produce wool. And it is also called as lanolin, wool fat. And here, the fat produced around the wool is called as wool fat or lanolin. What is its function? It is the coating of wax around the wool to prevent the water, to make it waterproof. And next one is Waxidi. This Waxidi is produced by a bacteria called Mycobacterium. The Mycobacterium species, they include Mycobacterium tuberculosis, Mycobacterium leprae, etc. This Mycobacterium tuberculosis that produce a chronic lung infection called as tuberculosis or TB. And whereas the Mycobacterium leprae species, they produce a leprosy disease. And here, this Mycobacterium species, they produce a wax called as Waxidi. This Waxidi, it confers the pathogenicity of the, because of this Waxidi only that conferred the pathogenicity of that bacteria. This is about the waxes. And now next we will see about our conjugated lipids.